Hi, this is Miss Litton, and this is my some of them wonderful zero period. Say hi. Hi. All right, so I wanted to record this last part about biochemical evidence and the cladogram, which you guys will be doing on Tuesday, but other students are doing on Monday. So this last part is the one that's used the most now, because we have the ability to make comparisons between DNA, and not only between DNA, but what does DNA code for? Proteins. And what are proteins made out of? Amino acids. So you can compare the DNA, and what's DNA made out of? Nucleotides. What are nucleotides made out of? Nitrogenous bases. What else? Phosphate, sugar. So the phosphate sugar part stays the same. The part that varies is the sequence of the bases. Yeah, the nitrogenous bases. So you can compare either one of those. And the idea is, the more similar the sequence of bases, or the more similar the sequence of amino acids in our proteins, the more what? Closely related, okay? And here you're looking at shared DNA sequences between chimpanzees and gorillas and orangutans, and where you can see differences indicate mutations. And those mutations could have led to adaptations. So that's another thing that you can compare, is the number of differences. If we have fewer differences between us, right, fewer differences would also mean that we share a more recent common ancestor. So that's what's referred to as the molecular clock, comparing DNA sequences to estimate phylogeny. Phylogeny is who you're related to, looking at that evolutionary history over time. So key things I want you to see here are the differences okay. The key things I want you to see here are the differences between genes. Indicates that you have a mutation. The more mutations that have accumulated, the more time has passed since divergence. And this is something you're going to be doing on Tuesday when you do your POGL. And then the rate of mutations are affected by many different factors. So take a look on your notes underneath biochemical evidence. Almost all living organisms use ATP, DNA, and specific enzymes. The more similar the DNA/proteins, the more closely related. So almost all living organisms use ATP, DNA, and specific enzymes. The more similar the DNA and proteins, the more closely related. Molecular clocks use comparisons of DNA sequences to estimate phylogeny and the rate of evolution. The differences of genes indicates the presence of mutations. The more mutational differences, the more time has passed. The more mutational differences, the more time has passed. Blue, tell them B. Go ahead, Blue.
The rates of mutation.
All right, so what we're going to be working at, we're going to look at phylogenetic trees, and phylogenetic trees can look like this and just kind of keep branching out. If we put them on a slant, I'm going to show you a little bit different. It's called a cladogram, and that's what your ed puzzle is going to be about, is how you set up a cladogram. And just a couple of terms I want to go over with you. The first part is systematics. Blue, tell them Blue just went?
Yeah.
Slate, tell them the definition of systematics. Go ahead. All right, so you can look at a lot of different things to decide who's related to who. You know, does it look like a bird? Does it have feathers? Is that important? Does it walk? Does it have mammary glands? Is that important? How many legs does it have? How does its development go through? Those types of things. You can look at how it interacts as a population. All those things can be used. And now, Slate, Blue, tell them the definition of phylogeny. Short-term memory loss. Go ahead.
Evolution is like evolutionary history of where it evolved from, how it evolved. Like a
Now, without me telling you anything, this is called a cladogram. See if you and your bio buddy can figure out how are cladograms set up. What do things indicate on this picture? Talk about it with your bio buddy on your own. Before I all right. So where you have these nodes right here, this is like the last common ancestor between hagfish and all the rest of these guys. But hagfish got left off because they don't have what? Jaws. From here on up you have jaws. And you get to be in that group. They all have something in common, jaws. Okay? Then as we move up here, then we have the perch that's sitting up here, but he doesn't get to move up with everybody else because he doesn't have what? Lungs. Because from here on up, all of these guys have lungs. And then you're going to drop off more because from here on up, all of these guys have what? Claws. And then from here on up, these guys all have fur and mammary glands. So what you end up doing is you get a list of all these attributes and you look to see what do they all have in common and who has it. If they all, and then you can put them all together on a line, on a cladogram, but then you have to decide who comes first, who do we start dropping off. Well, all of them had everything except one guy didn't have what? Jaws. So we dump him off. So he goes at the bottom. And then you take everybody else up and you start to put them in sequence. When you look at characters to put them on the cladogram, you can look at all different things. You can look at the number of chromosomes. You can look at structures and features that they might have. And you can also look at molecular features. These are harder to see because you're not visualizing like beak, no beak, mammary glands, no mammary glands. You're looking at has the fourth amino acid in cytochrome C or doesn't. Okay, so, but that is what's being used today, is you're developing cladograms not based on features that you can see, like that ugly thing hanging over his beak, which is highly attractive to the female, but you're not looking at those type of features, you're looking at sequences of DNA and setting up your cladograms. Okay, so a couple of things. This is called cladistics. It's a system of classification. And you look to see when did you diverge from that common ancestor. So you will look at ancestral characteristics, those that everybody had, right? All of those features, everybody had at least what? They didn't have what? There's only one guy who didn't have what? On when we looked at jaws. Yeah. Okay? And then you look at derived characteristics. These are things that come later. Like later, if we look back, okay, not everybody started out with claws or nails. That evolved later. So you put those guys next, farther up this chain. And you can see like I already kind of went through this with you. So I think you understand here, wherever you have these points, that's where you have your last common ancestor that you shared. Okay, I think we'll be okay with that. I'm going to move on. And I'm going to actually move on. We're going to practice that on two. Let's take a look at this. Okay, here we have some creatures, and we want to look at the way they possibly evolved. And here are some different traits. So where do we want to start? What do they all have in common, do you think? Backbone? Mm-hmm. So from here up, they have a backbone. Okay, what about next? We're going to pull fish out right here. Okay, then what are we going to put? Lungs, okay. So everybody from here on up has lungs. Now what? Okay. Claws and nails. Okay. Then what are you going to go with? What? I didn't hear what you said. Oh, the lizard. I'm looking for a structure. Okay, gotcha. They all have hair? Okay. Then what are you going to go with? Mouse? Opposable thumb?
Oh, that's, I thought
This? And then what are you going to put?
Bipedal.
Is this how you did it? Okay, so that is setting up a cladogram. So what the deal is you would, if you go back here, you would go, what do they all have? Do they all have a backbone? I did it that way because I thought you'd find that easier. You'd say, yeah, they all have a backbone, so that's going to go on the bottom. Okay, who has lungs? Everybody but that, right? Who has claws? And so what you do is you arrange them. Finally, at the very end, the only one that was bipedal was the human, so you know that's going to go at the end because he's the only one who has that, because we kept dropping off everybody else as we went along. Okay, that's how you set up a cladogram. But see, that's easy because those are things we're familiar with. But what scientists use is they look at sequences in amino acids and they count up how many differences there are. And so they go, okay, here's a human, here's a monkey. Let's see, we match. G, L, E, C, D. Do you see how we're looking to see that we match? And you count up how many differences we have. So here on this human, we have T, but a monkey has what? A. That's one difference. Okay? And so then you build your cladogram based on the number of differences. So it would be the same idea here, but we would say only one difference separates us between this one and this one. Do you see what I'm saying? And then here, there were 10 differences. If there are 20 differences, if there are 40 differences, if there are 60 differences, that's what's separating us. Okay? So that would be more a modern way to set up your cladogram. And then here you can see this as well. So you're using biochemical evidence to set this up. So here they're looking to see genes and who has what gene and the order of the bases within that gene to set up their cladogram. Okay, so on your notes, put phylogenetics is a modern way in which organisms are classified and arranged in evolutionary trees. When a new trait evolves and the tree branches, this is visualized as a cladogram. A cladogram reflects the evolutionary history / relationships. You have a common ancestor and all of its descendant lineages is called a clade or a clade. As characters, if it's an ancestral character, those that are found within the entire line of descent. Those that are found within the entire line. So for instance, in our entire line that we looked at, what did everybody have? Backbone, okay? Whereas a derived character, those are present in members in one group of the line, but not the common ancestor. But not the common ancestor. Okay, and we hope you understand cladograms better. Yay, have a piece of toast.